Thanks for a great presentation. Let's move on to our second presentation, which is Dr. Pittman's research group.
So we looked at conditioned and generalized avoidance of a variety of sweet stimuli following conditioned taste aversions to glucose or fructose. I'm Ashlyn Fox.
I'm Olivia Miller.
I'm Allie Newman.
I'm Amita Patel. And I'm Emma Pyle.
So the first thing that we looked at was sweet taste discrimination. There are 3 different receptors following this, the T1R1, the T1R2, and the T1R3. These receptors express certain taste buds and when combined in different ways mediate the sweet taste. The concentration of a sweet stimuli is compared to another sweet stimuli which affects the discriminatory ability. This is an issue because when combined with other receptors which lead to a loss in discrimination ability between sweet tastes. So there is a behavioral discrimination when it comes to this. It usually is determined if there are different sets of taste receptors for each taste sense. So in animal studies, it's shown that glucose or fructose are most common sweet stimuli used. This is because they are the biggest preference as well as how they are the common molecules that build up other sweet compounds. Next slide.
Like Ashlyn mentioned, we looked at types of sugars. We specifically looked at glucose and fructose since they have similar chemical compounds. And since they're so similar, it's hard for the rats to discriminate between the two. Glucose is important in maintaining sleep-wake cycles as well as hunger and satiety. And the role that glucose plays in the body is important because it regulates brain activity as well as it's preferable for them to taste it. While fructose has the same chemical compound, it doesn't affect the body in the same way because it doesn't go straight into the bloodstream like glucose does. It's converted to glucose later on in the liver. We also looked at the preference of sucrose and maltose. Sucrose and maltose are preferable to rats because they're sweeter in nature. Sucrose specifically because there are more receptors for rats to discriminate. And maltose doesn't have nearly as many receptors as sucrose, but rats still prefer it because it's so sweet. Because of this, rats are more likely to differentiate between sucrose and glucose and fructose because of the increased number of receptors that they have. And we use this background information because we wanted to use taste aversions for glucose and fructose to see conditioned aversions and generalized avoidance. Next slide.
So our study is based off of conditioned taste aversions. So I designed this little graphic here to explain conditioned taste aversions in a more simple way. So we had a group of 16 rats, and half of the rats received a saline injection and half received a lithium injection. So as you can tell in the picture, the rats would drink a fructose or glucose solution, and then the first group would receive a saline injection, and that's our control group. And then this wouldn't cause a reaction, so they wouldn't get sick, and they would continue to engage in licking behavior when the solution was presented again. And then in the second group, our experimental group, they received a lithium injection, and this caused them to be sick. So they would associate the sickness with the solution that they had drank previously, and they would decrease in their licking behavior. So next slide. So in order to gather our data, we used the MS-160, which is also called the Davis rig. And all of the rats were on a 23-hour water-restricted schedule before they were tested. So our Davis rig was set up to deliver 6 taste stints as well as a sample of deionized water. And as you can tell, the first picture on the right, there are little vials. I think, yep, there's all the vials. And each of the vials contain the different taste stints. So they would be presented to the rodent that's in the Davis rig, and there's a shutter that opens and closes based on how long the trials are. So our trials, the samples were delivered at a 15 or a 30-second trial. The Davis rig itself analyzes licks, latency of the first lick, as well as rate of looking for each concentration. The Davis rig is put into a isolation chamber that's covered in insulation, there are fans as well as white noise machines, and all this is done in order to reduce, in hopes of reducing olfactory cues. So I'm going to talk a little about our procedure. And so first, the rats had to go through a training period. And during this time, no injections or tastants were given to the rats. Only deionized water was presented in order for the rats to learn their licking behavior, or, kind of know what they're supposed to do while they're in the Davis rig. So next, we did the conditioned taste aversion test. During this test, it included 2 CTA pairing days as well as a single CS-US pairing day. So our rats were given access to the solution. After this, they were given injections, and then the following day, the rodents will be tested for the appearance of the aversion. So we also did a generalization test, and this is when the rats were tested for the opposite CS, and this was to see if the rats could distinguish between the glucose and the fructose. So later on in our experiment, we actually tested generalization to other sweeteners that we talked about earlier, like sucrose, maltose, and saccharin. So after we did all of the testing, we gathered our data from the Davis rig and completed a one-way ANOVA. And then we also did a post hoc t-test in order to look at between group interactions. So next slide.
So if you look at the graph on the left, these show the consumption of the unconditioned stimulus, whether that be glucose or fructose. It also splits even further to see whether the rat was injected with the lithium chloride or the sodium chloride. On the first day, none of the rats had been made sick, so they consumed all at an equal amount. But by the next week, they had been made sick, so we can see over time as the red and purple groups continue to be given the conditioned taste aversion, their consumption goes down due to continuously being made sick. The graphs on the right showed the trials in which we use a 30-second duration for each trial, and there were only 5 concentrations of glucose or fructose and 1 of water. So from the left to the right, it shows from day 1 to day 3. The top set of graphs look at glucose as the unconditioned stimulus, comparing the rats with lithium chloride injected to those with sodium chloride. So as you can see, as the days go on, these lines get closer to one another at higher concentrations. The red line being lower shows a conditioned taste aversion, which can be seen on day 1 at the strongest. And on the bottom set of graphs, we looked at fructose as the unconditioned stimulus, and a much larger difference was seen between the lithium and sodium chloride groups. So day 1 shows a conditioned taste aversion starting at an earlier concentration, and throughout the days it grows to a larger concentration. Next slide. So the remaining graphs use a 15-second duration for the trial and 6 concentrations of the unconditioned stimulus along with 1 of water. So the top row of graphs is again looking at glucose as the unconditioned stimulus, and as you can see, in day 1 there is a noticeable conditioned taste aversion between the 2 groups, and as the days go on, this goes away except at about 200 millimolar concentration. The bottom row of graphs use fructose, and between 60 and 100 millimolar is when the typical conditioned taste aversion is seen for these trials. Next slide. So we then changed into our generalization procedures. The left graph uses glucose as the unconditioned stimulus and looks at its generalization to fructose. The right graph uses fructose as the unconditioned stimulus and is looking at its generalization to glucose. Neither showed significant results, suggesting that when given a conditioned taste aversion to one, the other is different enough that the conditioned taste aversion does not generalize to the other stimulus. Next slide. So we then did the generalization procedures with other sugars. The 2 graphs on the left use glucose and fructose as the unconditioned stimulus and look to see if sucrose would generalize to either. Both of these had significant differences, and fructose had a noticeably larger difference. This suggests that sucrose does generalize to glucose after 30 millimolars and generalizes to fructose even more and at less than 30 millimolar concentration. The top 2 graphs looked at generalization to maltose. Neither of these showed significant results, meaning maltose does not generalize to either glucose or fructose. The bottom 2 graphs look at the generalization to saccharin. On the left is looking at glucose as the unconditioned stimulus, and there was no generalization, but on the right, there is with fructose. One explanation could be the way that the artificial sweetener saccharin binds to the sweet receptors is similar to fructose, but not to glucose. So this being the rats can tell them apart, can tell glucose and saccharin apart because they are nothing alike in their receptors. Fructose and saccharin cannot be differentiated at the receptor level. Next slide. In the avoidance test of the conditioned stimulus itself, the avoidance was greater for fructose when it was a conditioned stimulus as opposed to when glucose was a conditioned stimulus. The glucose unconditioned stimulus is what caused the rats to get sick, and because they generalized to fructose, it can be interpreted that the two sugars are perceived differently. Saliva in the mouth could cause sucrose to break down into fructose and glucose, which are the molecules that make up sucrose. Fructose is a more salient stimulus than glucose, which explains why the rodents were able to make the distinction and avoid it more strongly. Limitations of the study include the length of the study and problems with generalizing to humans. The study was conducted over 9 weeks, which creates the possibility of rodents experiencing fatigue. Extending the study with more days off in between each condition set may help with this. Also, including a trial time between the two used, such as 20 seconds, would help determine if rodents can taste low concentrations without too much exposure to the tasting. Future research may consider the generalization of other tastes. For example, would the results be the same if rodents were exposed to sour taste and then given lithium chloride? It would be interesting to see if there's a stronger aversion to tastes rodents already may not like and whether they are able to recognize these tastes at a lower concentration. Next slide. Thank you. We would like to take a moment to thank Dr. Pittman and everyone else for watching our presentation. And if there's time for questions, we would love any.
All right. Does anybody have any questions? Thank you. Thanks. We have time for questions if anybody has questions for this group. Anybody? No? Okay, how about a round of applause for that excellent presentation?